Good afternoon. My name is Prajwal Paudel, Product Development Scientist at ACROBiosystems. Today I will be discussing the remarkable progress in vaccine and therapeutics against COVID-19 at a record pace, and how ACRO has been intimately involved in partnering with researchers and pharma companies at each step of the way. The novel COVID-19 has to date infected 90 million people and led to 1.9 million deaths worldwide. The viral agent of this pandemic, SARS-CoV-2, is an enveloped virus with a single-strand positive sense RNA genome. SARS-CoV-2 is part of the beta coronavirus family and comprises of lipid bilayer containing membrane protein, envelope protein, nucleocapsid protein, and the spike protein. Rapid advancement in understanding the structural underpinnings of SARS-CoV-2 infectivity and transmissibility has yielded promising therapeutics and vaccine candidates. Continual monitoring and study of viral mechanisms and impact of emerging mutations are key to turning the tide on this pandemic. Among all of the structural proteins, viral fusion protein plays a critical role in facilitating viral entry and, by proxy, virus genetic material into host cells. After entry, host cell machinery is hijacked for replication, transcription, and assembly of viral particle, propagating its spread. In particular, the thermodynamic barrier of merging enveloped virus membrane to host cell plasma membrane is overcome by the kinetics energy stored in the fusion protein. The fusion protein can be divided into three classes based on their structure. Class 1 is primarily comprised of alpha helices, while class 2 consists primarily of beta sheets, and class 3 contains a mixture of alpha helices and beta sheets. There are four known triggering mechanisms for fusion protein, including receptor binding, low pH, receptor binding followed by low pH, and receptor binding followed by protease cleavage. The spike protein of the novel SARS-CoV-2 is a class 1 fusion protein triggered by receptor binding and subsequent protease cleavage. The mechanism of class 1 fusion protein begins with the pre-fusion state, where the receptor binding subunit is clamped to the fusion subunit. Triggering of the fusion protein after receptor binding and protease cleaving enables the receptor binding subunit to move out of the way, allowing the fusion subunit to form a pre-hairpin with the target membrane via the fusion peptide. Subsequently, the pre-hairpin folds back, causing N and C alpha helix heptad repeats to form a 6 helix bundles, pulling the two membranes close through the close apposition, hemifusion, and the fusion pore formation. Prior to SARS-CoV-2 outbreak, we had a smaller outbreak of the then a new novel coronavirus, SARS-CoV, in 2002. Much of our understanding of SARS-CoV-2 is due to the plethora of research on SARS-CoV. Although the four structural genes of the novel SARS-CoV-2 share more than 90% in sequence identity with SARS-CoV, the spike glycoprotein diverges with only 77% sequence similarity. The S protein consists of the S1 and the S2 subunit. The S1 subunit contains the NTD, or the N-terminal domain, and the receptor binding domain, or the RBD, which binds to the human angiotensin converting enzyme 2, or ACE-2. The S2 subunit comprises of the fusion peptide, heptad repeats, transmembrane region, as well as the cytoplasmic domain. The high transmissibility of SARS-CoV-2 compared with SARS-CoV has been posited to occur due to the insertion of a polybasic cleavage site between the S1 and the S2 subunit, comprising of proline, arginine, and alanine residues. The polybasic cleavage site between the S1 and the S2 subunit reduces stability of the S protein and facilitates conformational change required to bind the ACE2 receptor. Now, underlying the importance of this furin cleavage site, experimental evidence indicates the critical nature of protease cleavage prior to viral entry. Lentivirus presenting S protein in a pseudoviral entry assay shows that protease cleavage of S protein occurs during viral entry, mutating the putative polybasic cleavage site on S protein abrogated the cleavage. Furthermore, viral entry efficiency in ACE2 expressing HeLa cells and lung cancer cell lines was significantly lowered in cleavage site mutant of S protein. These results indicate the necessity of cleavage of the S protein for viral entry. The spike protein is intimately linked to the infectivity of SARS-CoV-2. Structural analysis of the spike protein shows a trimeric structure with a central stalk consisting of a 3s2 subunit, shown here in cartoon form, that is covered at the top by S1 subunit, shown as surface representation. The S1 subunit is dynamic, with conformation changes between open and closed. In the S1 open conformation, the receptor binding domain is accessible to binding via ACE2. These movements and rearrangements by RBD and NTD domains of S1 subunit are driven by energetics of ACE2 binding. ACE2 binding alters the open RBD conformation by a rigid body rotation of the domain, moving it 5.5 angstrom further away from the trimer axis, while the NTD domain moves 1.5 to 3 angstroms. The RBD associated subdomain 1 moves about 8 angstrom, while the NTD associated subdomain 2 moves by 3 angstrom. The NTD associated subdomain 2 interacts with short helix and loop of S2 subunit via a series of pi stacking in the closed conformation. These interactions are lost in the open conformation, leaving a channel in the S2 that is disordered and exposed for the next step of membrane fusion. The major rearrangement of the S trimer is fueled by the energetics of ACE2 binding events. ACE2 binding favors the RBD open conformation. As you can see here, sequential opening and binding of RBD in the S trimer leads to a fully open state, which leads to a substantial reduction in contact between the S1 and the S2 trimeric core. The fully open state leaves a 50 angstrom diameter around the trimer axis that is 65 angstrom deep. Furthermore, this new open structure leads to solvent exposure of the fusion peptide on S2, as well as the major helical rearrangement, which leads to host cell fusion. There are already multiple neutralizing antibodies in clinical trials targeting S protein to block the S protein ACE2 binding. Neutralizing antibodies isolated from COVID-19 convalescent plasma donors are roughly divided into 4 main classes. Class 1, 2, and 3 are all neutralizing antibodies, while class 4 antibodies are neutralizing antibodies that only bind RBD but do not block the ACE2 binding. Class 1 antibodies bind to open conformation RBD. Class 2 antibodies bind to both open and closed RBD conformation, and class 3 antibodies bind outside the ACE2 binding site. Importantly, the Regeneron 10933 and Regeneron 10987 antibody mix have received the FDA emergency use authorization against COVID-19 under the name casirivimab and imdevimab. Combination of antibody with the Fab region approaching at different angles to the RBD on the S protein is thought to be resistant to spike mutations and should be considered for development of antibody therapeutics. Recently, the NINDS reports of nanobodies against spike protein. These nanobodies are 12 to 15 kilodalton single domain antibody fragments derived from llamas. Nanobodies can be an effective therapeutic tool against COVID-19 as it is inexpensive to produce and can be delivered via inhalation. ACROBiosystems is proud to have partnered with researchers at NINDS to provide our high-quality S proteins to immunize llamas and test lead nanobodies against S protein RBDs. We provide both ACE-2 and RBD proteins for screening and isolations of lead nanobody candidates, as well as RBD mutants for affinity testing. The lead candidate 112 showed high affinity to wild-type RBD and maintained activity in blocking high-affinity RBD mutant variants binding to the ACE2 receptors. ACROBiosystems partnered early on with the development of Pfizer-BioNTech BNT162b1 mRNA vaccine as well. Early phase 1-2 safety and efficacy studies showed robust immune response after second dose at 30 microgram. High levels of anti-RBD IgG in sera were detected, and the serum displayed robust neutralization activity on par with COVID-19 convalescent plasma. Ultimately, Pfizer-BioNTech moved ahead with the full-length S protein 2 proline mutant mRNA vaccine BNT162b2 for their phase 3 studies due to better reactogenicity profile and stronger CD8 plus T cell response. And in last December, the vaccine was approved for against SARS-CoV-2 infections. The S protein RBD used in this study is expressed from HEK293 cells and isolated to 95% purity. ACROBiosystems has a proprietary biotinylation technology where the protein is biotinylated at the C terminus via enzymatic ligation of biotin to the AVI tag. ACE2 binding ELISA shows our product having strong binding to RBD with a linear range of 2 to 78 nanogram per milliliters. Now, closer examination of S protein structure reveals that domains in the S1 subunit and S2 subunit comprises of centroids and various angles and dihedrals. This means mutations at sites intersecting these centroids will affect the conformation landscape of the S protein, impacting vaccine and therapeutic development. In fact, to generate vaccine and therapeutic antibodies, pre-fusion stable S proteins are needed to increase recombinant protein expression with homogeneous preparations. 6 proline substitutions have been identified to stabilize loops and cap helices in the pre-fusion state, as well as introduction of disulfide bonds to prevent conformational changes. In addition, natural mutations outside RBD domain can significantly alter the conformation of viruses, which needs to be monitored continuously for any effect in transmissibility and severity of COVID-19. Therefore, ACROBiosystems provides a super stable S protein trimer for both vaccine and therapeutic development. The trimer contains a T4 fibritin trimerization motif in addition to the 6 proline substitution to stabilize the trimeric prefusion state. Additionally, the furin cleavage site has been mutated via 2 alanine substitutions. We have isolated high purity S trimer with high homogeneity. The S trimer particle structure was verified via negative stain electron microscopy and matches the structure reported in the literature. Additionally, performance testing of our superstable S trimer with wild type S trimer, the 2 proline substituted S trimer, along with S trimers from competitor, showed ACRO's superstable S trimer with 6 proline mutations to have consistently have the highest signal to noise ratio. Such high performance will greatly improve development of diagnostic kits as well as vaccine immunogenicity evaluations. As SARS-CoV-2 spreads globally, random mutations of viral genome is inevitable. GISAID is tracking new mutations or clades of SARS-CoV-2 in the course of this pandemic. Next strain visualization of the SARS-CoV-2 mutation shows multiple clades of mutant emerging in 2019 and 2020. However, mutation that leads to the gain of functions are especially dangerous as it increases viral transmissibility and potentially disease severity. To date, the D614G variant is the dominant mutation found in virtually all SARS-CoV-2 infections. Recently, 2 new variants have emerged, one B117 from UK and the other 501Y V2 variant from South Africa, and both had mutations in the RBD of S protein. An early mutation, D614G, is now virtually present in all SARS-CoV-2 samples. The mutation causes loosening the S trimer structure via abrogation of this hydrogen bond between the D614 and threonine 859 residue on the adjacent protomer. This leads to a shift in equilibrium of the S conformation from 47% open to the 95% open, either 1, 2, or all 3 RBDs in the mutant trimer. Thus, this mutation increases infectivity of SARS-CoV-2, but however, it does not appear to affect the current vaccine efficacy and neutralizing antibodies targeting the RBDs. Another recent mutation is the B117 mutant strain first observed in UK. There are 17 different mutations and deletions in the strain, but most of the mutations are found all around spike protein. The 69 to 70 deletion and the N501Y mutation are particularly of interest, as some evidence indicate a gain of function. This strain has quickly taken over London and UK area in the last couple of months and has been observed in multiple countries to this date. At ACROBiosystems, we have multiple S protein mutants, including the D614G strain, as well as the new UK B117 variant and the new South African RBD triple mutant variants. We also carry key deletions and mutations of the UK variants, particularly the 69 to 70 deletion in the NTD of S1 subunit, as well as the N501Y mutant on the RBD of the S1 protein, S1 subunit. Additionally, ACROBiosystems also offers single mutant variants of S protein RBDs. The rise of the mutants, particularly the new UK strain, poses concern in the efficacy of recently approved vaccines and antibody therapeutics, and ACROBiosystems is proud to provide researchers with these mutants for rapid discovery of any effect in vaccines and antibody therapeutics. Now, in the diagnostic space, ACROBiosystems has developed multiple serological tests to track and fight the surging COVID-19 spread. Testing the levels of the neutralizing antibodies against S protein serum in samples is critical in determining the efficacy of vaccines as well as diagnostic measures of past infections. With the beginning of a large-scale immunization with multiple vaccines and the rise of mutant variants, close monitoring of neutralizing antibody in population and the impact of virus mutation is necessary for effective immunization campaign. ACROBiosystems offers neutralization kits as well as IgG, IgM titer detection kits. Our neutralization kit showed consistent detection of neutralizing antibodies against various dilutions of COVID-19 convalescent serum. ACROBiosystems serology kits enable better understanding of neutralizing antibody mechanisms and help researchers develop strategies to improve vaccines and diagnostics, particularly against emerging SARS-CoV-2 mutant strains. To understand these effects of the mutations on RBD of the S protein and the resulting impact on the ACE-2 binding, we can examine residues that are involved in these interactions. There are 5 key residues on the RBD that make physical contact with the ACE-2 receptors. The key interactions are similar between SARS-CoV and SARS-CoV-2 mediated by polar residues. Particularly, an extended loop region of the RBD in the S protein forms a bridge with the alpha-1 helix of the ACE-2. Current mutation of concern within the RBD is the asparagine 501 to tyrosine, present in both the UK and the South African variant. In molecular dynamic simulations, shows the tyrosine residue, mutant 501 residue, forming a pi-pi stacking with the tyrosine residue 41 on ACE-2 and coordinating with the hydrophobic, through hydrophobic interaction with lysine 353 residue on ACE-2 as well. Now, this mutation is estimated to improve binding by 0.8 kilocal per mole. Further study is needed to understand the effect of this mutant in RBD ACE-2 binding. Now, examining the ACE-2 receptor structure closely is also critical in understanding the high transmissibility of SARS-CoV-2. Biologically, ACE-2 functions to counteract the renin-angiotensin system to convert angiotensin peptides, which are hormones regulating vasoconstriction and vasodilation in the cells. The ACE-2 receptor is found in high levels in the heart, kidneys, and testes. ACE-2 structure consists of the functional extracellular peptidase domain, followed by the collecting-like domain or the NEC domain. Then the transmembrane domain follows, and a 40 residue intracellular segment not resolved in these models. A protease cleavage site in the NEC domain of ACE-2 enables secretion of active ACE-2 peptidase in the extracellular matrix. The ACE-2 dimerizes mediated primarily by the NEC domain via hydrogen bonding and cation pi interaction into an open and a closed conformation as shown. The open conformation binds to the RBD domain of the S protein as shown. Docking the S protein trimer reveals that each ACE-2 dimer can bind to 2 S protein trimers simultaneously. Now, it remains to be seen whether clustering of S trimer and ACE-2 dimer occurs and enables fusion of entry of SARS-CoV-2, but ACROBiosystems offers the human ACE-2 as well as mouse, macaques, rat, and civet ACE-2 for further study. ACROBiosystems also offers ACE-2 with a variety of tags, including HIS, AVI, and FC tags, to facilitate in-depth study of S protein ACE-2 binding mechanism and assessing efficacy of vaccines and therapeutics. Now, in addition to ACE-2, neuropilin-1 receptor, which binds to furin cleaved substrate, has been shown to facilitate SARS-CoV-2 viral entry, particularly in the nasal cavity cells. Expression of both neuropilin-1 and ACE-2 receptors in HEK293 T cells significantly improved viral entry than expression of ACE2 alone. In fact, due to low levels of expression of ACE2 receptors in the respiratory and epithelial cells, neuropilin-1 receptor is thought to promote entry of SARS-CoV-2, enabling cells, vascular, and tissue penetration. ACROBiosystems offers His-tagged and biochelated neuropilin-1 with high purity and bioactivity. Recently, the S1 domain of SARS-CoV-2 has been shown to bind to cholesterol and components of the high-density lipoprotein. Therefore, the HDL scavenger receptor B type 1, or the SR-B1, has been implicated in enhanced ACE2-dependent viral entry as seen in this pseudoviral entry assay. Expression of SR-B1 improves viral entry, while knockdown of SR-B1 worsens viral entry. SR-B1 is co-expressed with ACE2 in pulmonary tissues and acts as a host factor promoting SARS-CoV-2 entry. ACROBiosystems is proud to partner with researchers at Beijing Institute of Biotechnology in uncovering this mechanism of this novel interaction, which can be a novel therapeutic target. We are excited to continue partnering with leading SARS-CoV-2 researchers in elucidation of the mechanisms of viral infections and develop new therapeutic strategies. ACROBiosystems currently supplies recombinant proteins, including mutants, antibodies, kits, and pre-coupled beads in the fight against COVID-19. Please visit our website at www.acrobiosystems.com for more information and to learn more about our more than 1,600 recombinant protein catalog, including immune checkpoint proteins and CAR-T targets. ACROBiosystems is ISO certified, and some of our COVID-19 diagnostic kits are also IVD CE certified. My email is prajwal.paudel@acrobiosystems.com for any questions you may have. For any general inquiry or product information, you can contact us at order@acrobiosystems.com. I would like to thank the internal PD team at ACROBiosystems in developing our COVID-19 catalog and addressing many technical hurdles to provide high quality and high bioactivity proteins. We are immensely proud of our pharmaceutical partners and enabling their successes in developing novel and effective therapeutics. We are grateful to our partnership with research labs and institutes for working closely with ACRO in exciting discoveries and to improve our product lines. Thank you for your attention and participating in our talk. We are actually doing a raffle for our llamas, so please come visit our booth to enter the raffle and have the chance to win an adorable and plushy ACRO llamas. And with that, thank you for your attention.